Okay. Welcome everyone to the very first Monash Partners Comprehensive Cancer Consortium Tech Talk. My name is Thu Nguyen Dumont and I'm a researcher at Monash University. I would like to begin by acknowledging the people of the Kulin Nations, who are the traditional owners of the land on which we are gathered today at the MPCCC and across Victoria. I respectfully acknowledge their elders, past, present and emerging, and I pay my respects to any members of the Aboriginal and Torres Strait Islander community gathered among us this morning. Together with Professor Melissa Southey, Chair of Precision Medicine at Monash University and Research Director of the MPCCC, and Vicky Marshall, Program Manager of the MPCCC Precision Oncology Program, I would like to welcome you to what we hope will be a beneficial morning of discussion for researchers and clinicians. MPCCC Tech Talks are an online forum we've developed to facilitate discussions about how new technologies can be applied in the lab or in a clinical setting. We'll be inviting researchers and clinicians to talk about their experience using novel technologies, the advantages and disadvantages of using one approach or another. And we hope this will be a valuable opportunity for our community to exchange and gain insight into novel analytical and technological developments. So we're delighted to be joined by Dr. Renata Pellegrino, today live from the Children's Hospital in Philadelphia in the United States. Dr. Pellegrino is the Technical Director at the hospital's Center for Applied Genomics, and today she will talk to us about how she's used Twist BioScience solution for next-gen sequencing library preparation. Also joining us today is Jeremy Dunstan, who's the Sales Director of Twist BioScience. Renata and Jeremy will both be answering questions after Renata's presentation. Before I hand over to Renata, I would like to go over a few points of housekeeping for this event. Could all attendees please ensure you have your video turned off and your microphone muted before we begin? I'll be moderating questions at the end of Renata's presentation for both Renata and Jeremy. So to ask a question, please click on participants at the bottom of your screen and select raise my hand. Alternatively, you can also raise your hand physically with your video on so I can see you. And please remember to unmute and introduce yourself when you're asking your question. And after Renata or Jeremy have answered your question, please click lower hand and put yourself back on mute. So now, without further ado, allow me to introduce you to Dr. Renata Pellegrino. Renata has over 15 years experience in genetics and genomics research. She's responsible for the Genomics Lab operations at the Center for Applied Genomics. And she's going to tell us about her experience applying Twist BioScience in her lab. Welcome, Renata, and I'll hand over to you now.
Thank you.
I'll just let me unshare. Unshare. Oh, I lost my Zoom window.
I think I can share already, I think. Oh, no, I can't because you're sharing.
Oh, thank you.
You got it? No. So can you hear me well?
Yes.
Awesome. All right. Well, thanks for this awesome invitation. I'm very happy to be here virtually with all of you. We are quite friend of Zoom and GoToMeeting in the past months with COVID, so this is quite interesting new, life for all of us. I'm assuming the same way for you there in Australia. So I would like to also first apologize if you get some interruption with a dog barking. I have my dog and she's very needy, so I really apologize if she barks or whines for me. I was trying to not give her attention enough. Tonight here in Pennsylvania, today is morning already in Australia, of course. I'm going to talk about these advanced technologies and how this open new horizons in pediatrics, but I don't think this is restricted to pediatrics, because I think this has been applied to many other, scenarios in adults, and also in agriculture and in other areas in genomics. So, and how we work really well with these new technologies, but making partnerships and like having this fantastic partnership with Twist in the past years now. So I'm going to tell a little bit of a story, how we develop some products together and how I create some tests in my lab that it's extremely well implemented now. Okay. So a little bit of my city here. I am not from Philadelphia. I'm from Brazil, actually, but I live in Philadelphia for many years, and I absolutely love the city. So it's a quite popular city in the country, United States, and it's very popular for some individuals such as Rocky Balboa, Benjamin Franklin, but also many movies, and it's a very diverse and happy city overall. So if someone wants to come and visit my lab and my city, I feel very happy. So this is my this is the Children's Hospital buildings. We have like multiple buildings, and we are the largest pediatrics facility in the United States, and we are one of the largest in the world. So we occupy a lot of space in the city. So the city is not that big. So I used to say that this is like a view that you can tell we are quite a big hospital. A little bit of background in my lab. So the lab it's about 12 to 13 years. Now we have about 80 people working for us. The PI is Dr. Håkon Ersson, and I am the technical director for the entire operations, the wet lab related operations for the center. So the center started, as I said, 13 years ago, with this vision to become the largest biobank for pediatrics. At that time, we had no idea that that would be the case, but that's how it started. And we create a very sophisticated way to do that. So we start collecting samples with the intention to also genotype those samples. And we evolved to phenotyping in the course of our development. So I am part of this lab for 11 years. So I came like 2 years later when the lab started, but many things happened since that. And I have this highlighted here in red is the wet lab. So the wet lab has done million type of different experiments, but we became very popular to do GWAS this back 10 years ago. And we evolved to sequencing, and not only sequencing with like very regular technologies such as whole genome sequencing and whole exome, we start to adventure in RNA-seq, single cell, CRISPR, and a lot of functional studies. So we have a lot of functional because we do something called translational medicine, which is very, very, very strong in our lab. And we also have a very strong bioinformatics team that develop popular softwares that are free and available to everyone. And the final goal of the lab is not only to identify new variants or sequence massively number of individuals or genotype massively, but actually target therapies. We want to really see if we can find a variant that is actionable and we can have some impact in a patient lives. So we do have a very strong activities on rare disease, but also common disease. So if you go to this side of this slide, which is a little bit busy, but I want to just mention that we have a very diverse population of phenotypes and disease, starting from our main cohorts when we start psychiatry, moving to autoimmune, cancer. The numbers are way bigger now. Every month I present this, and it's changing because we genotype constantly our biobank. So we have now about 400,000 genotype samples, including parents, and sometimes we also collect PBMCs that we can blood to provide PBMCs, and that we also immortalize with EBV transformation. So basically we have a very comprehensive biobank, and we became the largest biobank in the world for pediatrics so far. The center has this portfolio, as I just mentioned. So we have the NGS core, we have the BioBank genotyping core, translational lab, the team for bioinformatics, and we do have a new area, which is the CLIA certified products, which is pretty much a clinical lab that is operating for diagnostic if needed, because everything in the United States for FDA now for drug development has to be done in a clinical environment. So we got our own license and we are independent. We can run any FDA test development, drug development in our lab now. So the biorepository is quite interesting. Again, I apologize for a busy slide, but I think it's important to give you the full information. So as I just mentioned, we recruited over 450,000 patients so far, where we are collecting, again, DNA, blood for DNA, for PBMCs, and sometimes RNA. But we also have evolved in collecting challenging samples such as saliva, Ffp, and dry blood spots. So that's why I make a highlight here again in bold that we have processed over 12,000 blood spots, and many of those samples are going to NGS, which has been a very interesting challenge. So just again, I keep saying that this is how now moving to the NGS lab, how it works. So we have our division for RNA-seq, DNA, and we call land rental. We joke around in the campus saying that we do a bring your own library. Don't bring your own breeder. We cannot do that, but we can bring your library and we can sequence based on each project. We are a very, very high throughput lab. So we have this system here in red called RAMP. The RAMP is an underground robot that can fit this 10 million plated samples. So it's a very insane robot that picks up our samples, so we don't touch samples anymore. Basically, it's always stored in these compartments here, and we just program the computer and the computer gets our samples. So it's quite protected system, and only 4 individuals can access this robot now. So I hope nothing happened to one of us, because this is quite protected environment. We have multiple robots for processing our samples. And the reason I'm gonna mention the robots is not because I wanna say, oh, we're doing a robot, is that when you talk about library prep and specifically challenging samples and specific projects, not always robots are the best option. So our goal in the NGS R&D division is always to improve library prep from the bench to robots. So that's why we like to make an emphasis here on the robots. And like genetics is quite popular, right? So we talk about genetics all the time. And the reason why I like to use this slide is because I feel like we used to be far from like common community and people that were not in science. We were like so far this back in the age, and now we are so close. Like I can tell my mom, my aunts, my goddaughter call me and say, did you see that something blah 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 happened? And I feel that with that knowledge we have a very strong obligation to give right message, but also responsibility to really make sure patients are tested properly, because now the patients are much more engaged to understand the test, the disease, and how can be managed. Many of our treatments now can be guided by genetic tests. So I feel that we always have to make a pause and understand now that patients and the community are much more aware of what's happening. So a good thing about genetics in the past decades. And when you think about the human genome, right, we're going to talk about exomes soon. And I always like to remember that when we start this, we thought we had 30,000 genes in the human genome, and people thought we will have much more and end up that we only have that little amount of genes compared to other organisms. And this publication is from 98, so not even thinking about human genome, get ready. And another thing that I always like to remember is what is really next generation DNA sequencing. Like this publication is from 2008, and we are now in 2020, and everything happened and changed. So what is next really, right? So again, I keep asking this and kind of provoking. And this is another interesting publication saying 10 years of next generation sequencing, 10 years. And we are still talking about NGS as a very sophisticated, amazing thing, which it is, but it's a really next, or we are actually in the present now and enjoying what we know. And when you think about this as well, it, again, another publication from 2008, they kind of like plotted all the technologies that were ascending, and NGS was like coming there. And now I keep saying that we have so much other fancy things going on, right? Link reads, we have all this, single cell and approaching, and exomes, of course. So I like to keep this in mind. And I can hear some background. Okay. It's okay. When we see this slide, it's quite interesting too, because it's like the most interesting marketing slide from sequencing companies that I ever seen in my life. It's not from sequencing companies, it's from NIH, but obviously people believe that I really cannot speak because it's a lot of background. Are you guys mute? Okay, it's better now. Thank you. So when I think about genomes, I feel like we are super happy, excited, right? In 2018 and 19, they announced that the genome is going to really cost $1,000. So we started 100 million, and they announced like $1,000. So we are very excited because we could publish much more. We are celebrating. This is a Super Bowl, so Americans are crazy for football, right? So we Philadelphia Eagles, we won. So we were like completely crazy in the streets. So that year we are also happy because these announcements of sequencing come out. But in my opinion, that's the dangerous part of the whole genomes. I still think in the clinical settings, interpretation is going to dictate the problems. And okay, you have a whole genome, but majority of the findings you cannot really interpret properly. How you can do very good variant curation and filters and all that. So in a clinical setting, it's quite challenging compared to this companies offering whole genomes for $500 now. And if you go to AGBT, this meeting, it's quite shocking to hear them say, oh no, you can do now for $100. So I feel a little bit cautious and concerned. Doesn't mean that I don't like whole genomes, and I do a lot of whole genomes, but in the clinical settings, I still feel that our exomes are extremely powerful, or panels, or in that sense. And is the fact we want to put the puzzle together, and when we do library prep, we create a lot of problems, bias, and it's really hard to put the puzzle together if you don't have a good method and a good technology. So all this introduction is to start a little story with me, my lab, and Twist. So this is my city, São Paulo. Actually, this is really where I come from. It's quite overwhelming. And this is the main hospital in South America, and is a public hospital. And I have an affiliation with them since I was part of them as a researcher, collaborator. And I have these friends that they work with neurology and neuropsychiatry, and they have this very challenging epilepsy panel. I'm not a neurologist, so I don't understand epilepsy much, but I said, well, what do you exactly need to do? And they said, we need to build this panel And we try with other vendors here and it's not working. And I know you can do very magic library preps, which I don't think is that totally true, but I said, okay, I can give it a shot. I have very good R&D folks, so let's try. So they asked me to help them on this project. So the disease model was like, we have this patients from very remote area in South America with seizures and epilepsy events. And they were trying to look for these specific variants, not only, point mutations, but actually also copy number variation. And they were collecting these individuals with seizures starting age of 2. But the caveat comes down, this specimen. So they said to me, only problem here is because we have dry blood spot samples. And they're quite old. I'm like, it's okay. I know that these blood spots are very valuable and they are very helpful for the, newborn screen. But, like that's what I like to say, but degradation in low concentration always been a problem for downstream analysis with this type of specimens. So I'm like, that's fine. I can take care of blood spots. I do blood spots all my life. I can do 10,000 blood spots in my robots. But then I didn't realize what it was coming to me. And this is exactly what came to me. So this is a control. This number 3 is a control for my own lab, my own collection. And if you go to, for example, number 4, number 7, number 11, this is completely like very weird pattern for blood spot. So it was quite challenging. So we did take care of this. We worked with many vendors for DNA extraction. We did QC, and then Twist came on board. So Twist was a very new company. They had a very tiny, small booth in the American Genetics Conference, and I knew the folks from Twist from many years from former companies and development that I have known them in the past. And I was very excited to hear about it, but I was very also concerned. I'm like, this is really good. But they said, no, we can't design this. And I was so tired of other vendors like promising or over-promising. So I said, you know what, I'm going to give a shot to these people and see, because they're very smart and their strategy seems to be very good. So I accept the challenge and I designed a panel with them. And at that time, that's why it's very clear here, they didn't even have a library prep. They only had probes. This is back 2 years ago. And I said, that's fine. I can use other type of library preps and just use your probes, because the probes are the challenging portion. And also, I didn't even have a NovaSeq, which is the newest sequencer. I used to have a HiSeq. So then our pilot results was very encouraging. So we get a very high on target, and this was done in a robot with a very, very horrible DNA. Our duplicate rates were pretty reasonable, and our 20x was pretty much 100% covered. So I was quite encouraged to continue this work with them. So this is a picture of my team. So my bioinformatician and my R&D scientists that worked for me. And then on the pilot, we start to evolve our interpretation. And I have a PhD student now that she's working on this for her thesis, actually, because she came to train with me and she become more involved with this development and being in R&D. And again, I'm not focused on these genes. I'm not a neurologist, as I mentioned, but I want to give attention to the coverage and the distribution of the probes. So that's the most important thing for me in this scenario. Again, my dog, I apologize. So if you have here the distribution of the probes, you can see the exons are properly covered equally across the entire gene. So basically there's no drops. There is no, losing an exon. And this is quite important for many diseases, because if you have a dropout or something that is not covered properly, you most likely is going to miss a variant. So that was so important to us. So then we feel very encouraged. So we're going to go now for this new adapter. So Twist came with this new adapter and a full solution for library prep. So I was even more excited. I was like, perfect, because I don't want to keep mixing multiple vendors together, because it's fine for research, but again, in the clinical lab, it's quite challenging to control quality control and make sure everything's compliant from different vendors. So I was quite excited. And then that's what I come here. I say, when it's time to do whole genome or when it's time to do a whole exome. So I continue to be a fan of exomes. And exomes, as we all most likely know, it's pretty much the coding areas on a genome, but most likely the disease, the disease-related variants are in the exome region. So that's why we did that. And then, as I said, we did the adapters. We say, let's test this now with the new adapters and also on the exome panel. So they came with this beautiful design. So I have here some other vendors that I have been tested. And again, it's very also challenging for us to believe which is the best final design and content. Content is always controversial. So Twist has a very comprehensive content, and we have our own, I would say, enhanced, which is around 39 megabytes. But other vendors have a huge content, but sometimes they have more than 3,000 probes in pseudogenes that we will never resolve with short reads sequencing. So it's almost like helpless to have such a content if you really cannot benefit with short reads. So I always question that when I go to meetings and we discuss in panels about like when we do the design, because it's always questionable in terms of like what's the real content useful for certain disease. So this is the comparison that we have done. So we did use the universal adapters that are the new adapters. I call them super adapters because they are really amazing and they improve a lot of quality for me. And I also compare with the old full adapters. So I did the Twist exome universal adapters complete kit, and we did also new universal adapters compared to another vendors using the epilepsy. So we did exome and we did the epilepsy panel comparing the adapters. So two different questions, but in the same type of experiments. So this is our first shot with very obscure samples from paraffin. So these samples I had for years. We tried every single method in the market, 3 different companies, different conditions, never worked. I had a little tiny piece left and I was like, well, that's the last chance we have. And I was like very like not motivated, but we did. And the results were very outstanding. So this was like in the bench. We did not use the robot. I need to be clear here because it was an extremely rare sample. And we got very decent on target. When I looked for the full adapters, which is the blue, full adapters are regular adapters that are very popular in the market. They are from Twist, but they are very big length compared to the new universal adapters. And I have a very good coverage on 20x. For paraffin, this is fantastic. But the most impressive information here is the duplicate rate. So we have 3% and 4%. So it's basically nothing. For paraffin, it's very low. Consider the number of PCR cycles that we have to introduce in this experiment. But then when we test the new adapters, our on target jumps to 82 to 83%. The duplicate rates were very conservative, and our mean coverage significantly increased. So again, we have such a big improvement that we got so excited to start to do that in more like large scale. Then we also tested the full adapter and the new adapter again with our old epilepsy panel that we have being very successful only using probes before, but now we use the entire setting, and the results were already amazing. But then again, we just have a very impressive improvements that we were like the on target going really higher when we think about the new universal adapter. So the same for the duplicate rate. So again, proving that these new super adapters are really super adapters. And once again, I selected 2 genes here. Again, I'm sorry for the dog. And we see a very beautiful distribution of the probes across the entire gene and covering all the exons. And again here, it's quite similar, but definitely I feel that the uniformity, which is the magic word for Twist, is uniformity. So basically you don't miss much areas over sequencing or under sequencing. This is a very good example for uniformity on whole exome sequencing. So vendor A, A1, B. A is the same vendor, but different captures, and vendor B is another capture and this gene. So these are my comparisons for Twist. So I did a lot of comparisons using Twist. You can immediately see the coverage for Twist probes. And again, don't forget the design is very conservative. It's only 39 megabase. When I say only is because vendor A has a 54 megabase design. So you expect that a better coverage is gene, for example. This gene is a very important gene for one of my colleagues in my lab. So that's why I put that as an example. And then you see a huge gap in this area of the gene here, right? In this exon here, and some drops. But you see a vendor B performing reasonable, but not as good and uniform as Twist. So Twist not only have a better coverage across the entire gene, but also has uniform distribution. So it's very important. Again, I'm not spending extra money sequencing what I don't need it or under sequencing important regions. Again, because my mutation could be right here and I could miss if I was using this vendor A or A1. And then we get, again, more excited and we said, all right, this is all done in R&D level in the bench. Now we need to make this happen in a robot, right? This is the main challenge. So we have done paraffin, blood spots, regular blood, saliva. I don't have the data from saliva here, but fantastic data from saliva. And I feel like, okay, so when I compare this here in a wet lab perspective, I see vendor A fully automated too, but 5 days prep, 5 days on library prep, fragmentation, shearing, and sample input, it was really high. And sometimes we don't have DNA. I have a lot of patients that are deceased and babies and newborns that we just cannot withdraw enough blood and, and there's no DNA available. Vendor A1, the prep was not automated at that time, and 2 days prep, which is better, but sample input was questionable. Vendor B, same scenario, different, library prep, 2 days. Fragmentation was questionable. Hive was completely manual, so it was very painful and very dangerous for mistakes. And the input was decent. But then, here's the winner, fully automated. So now it's 100% automated. 2 days. Frag enzymatic. We want to do everything on deck on the robot. And sample input 50 nanograms. We now can go as low as 20 in our lab with good results and very low duplicate rates. So again, this was actually we used this table to guide the decision for the hospital and the center and our procurement to choose the best method. So that was type of table that we created. So our goals were to scale up without compromising the quality. So in a high throughput lab, we can just run 10 samples a week because we have a very high demand, not only as a core, but I have multiple clinical trials ongoing. And one of my clinical trials I need to deliver per week around 300 exomes. So I really need to, make this in a scale that I can not only make libraries, but sequence on time and deliver results to the bioinformatics team. So our progress is start with good ideas, but not sure if it's going to happen. And then we did create our custom design with the epilepsy panel. Then we were a little more, brave enough to say, let's do a whole exome now and test some super adapters and challenging samples, but also regular samples. And then we evolved even more, improving our content. So we say, let's add a little more in our design, finalize our automation, do now maybe fast hype. They have a fantastic fast hype, and use even more challenging samples. So we are now in a scale of 1,000 a month. So this was completed in 2019 and 2020, which I don't count anymore as a great year because nothing pretty much happened since March to now, but we are still running 1,000 exomes a month in our lab. And how this is very important. It's not only numbers. I like to say that I am not a core facility for genomics factory. We are a thoughtful genomics center, and our goal is to really help patients. And personalized medicine and precision medicine is very important to us, and we are learning and getting into that, like this enabling breakthroughs from benchtop to bedside. So literally we are trying to find answers that can be converted in treatments. It's not easy. It's not like a magic thing. It takes a while, it takes years, but that's our goal, the mission for the center. And talking about mission, I think it's important to say and give you examples of all the success with sequencing, with technology, with genomics, and how we evolve in a way that we really see now, we see the results in patients' lives. So we have here this very beautiful case. I'm a fan of her, Emily. Emily is the first patient in history to be treated with CAR T cells. And she was treated in my hospital. And we are part of the story, not because we don't produce CAR T cells, just make this clear, but we do a lot of sequencing for them, or we work in partnerships with many groups that are working the CAR T cell development and engineering, and we do sequencing for them. But the message is how technology really impact, and again, how we can discover this special areas on the genome that have an impact in immune response that can be used like we are doing now. So Emily is a survivor. She has now a foundation because it's really hard to get treated, right? It's a very multi-million-dollar treatment. It's very expensive, but she created this foundation to really trying to help fundraise money for other kids in her situation. Like she had a very aggressive acute lymphoblastic leukemia. So they basically, this is just a quick overview. This is the main publication that came from the CAR T cell, and that saved Emily. So basically you remove cells, you re-engineer the cells, and you put it back in a patient. Sounds very simple. It's not that simple. I've been visiting the lab that produced the CAR T cells, Dr. Ngs Lab, and it's a fantastic facility, and it's completely like crazy how beautiful that people work and how hard it is to make the cells. But again, I just want to give you examples. So this is another very important example. This is part of my lab. This was a publication from last year. So we found a mutation in a lymphatic disorder. And this child, he was very, very ill. He was basically, I would say, dying, very, very ill. And he came to us, and we literally run exome on that was the last shot for this. Many geneticists have seen this kid, and Daniel was like followed by other hospitals. So he came to Philadelphia, and we found this mutation. It was a gain of function, and it was a ARAF kind of ERK1 receptor. So when we found this, we spent a lot of time in my functional lab, in translational medicine lab. And the team spent about 8 months trying to do, cell transduce with ARAF and S21-4P to see if they could bump the activity. And they finally found one compound that could help and the drug that could be a target. So it's not a super different drug, so trametinib, and the patient responds. So this was like the Nature paper, and this is just an image to, again, this is not actually my field. I also don't work with lymphatic disorders, but it's a very easy to see that all the lymphatic system was extremely compromised, and it's a very severe inflammation, and they gain proportions that the organs stop to work because they are so overgrowth that they basically do not respond anymore to regular functions, and they have a very severe lymphedema. So you can see the picture of his legs. So he's a very slim child, and he was extremely lymphedemic. So when he was in intensive care, we barely could recognize him anymore. So he definitely responded to the drug treatment, and he's cured in a way that he still have a mobile ventilator. But he's running, he's happy, he's going to school, things that he was not doing for years. So it's a very big victory for us. So in terms of direction, I like to say that as a genomics person and scientist, I feel sometimes myself in this confusion, because we have so much competition, people, companies, industry, private clinical labs, everything new, startups coming, and it's a lot of information to digest, and it's nearly impossible to test everything, to understand everything. But definitely, I think it's our mission to, when I have the opportunity to do some work in development and R&D, be honest, transparent, and speak about something that is working properly in academia to benefit the patient. So I always feel like I am very careful to see all this information and how I really decide what to do. Again, I really rely on other colleagues and other lab directors to say, yes, we have been testing this, we have been approaching this, and this is working. I also like to say that I am obsessed with history, and I went to Egypt 2 years ago, and I was there for days and days doing all this crazy archaeological sites with a private guide. And what is this important for genomics? The importance comes now. Weeks later, when I came back, I thought I saw everything. I know everything about Egypt. I know all the tombs. I rocked. I know everything, period. And then days after I was here telling and bragging about my trip, they found a completely new tomb, just side by side of the tomb that I was. And I felt like, oh my gosh, why? And the message is, we do not know everything always, and there are always room for improvement and for new discoveries. So I learned my lesson that, and technologies are constantly changing and improving constantly. Market is very active. Education is very important. Invest time in R&D and be brave. If you have the funding, you always have to be careful, of course, but that's my main message here. But you have to be brave to really invest your time and staff if you have, of course. And everything we do, we look for translational medicine and improve diagnostic. And we also want to have time, because if you have to do library for 5 days, now I can do libraries for 2 days, maybe 1 day. Now with the fast hype, I can spend time with my, best friend. So why not to really improve the quality of your technology? And obviously, we always thank the patients first and their families, because without them, nothing happens. They are our motivation for sure, even more when I'm in a pediatric setting. So definitely they are my reason to do this every day. And it's hard to say thank you because again, it's a huge family. So this is me and my entire group and the main PI in the lab and other team members here. And this is like all of us. This is my direct team in the sequencing lab. I have much more people, but again, it's hard to, but I always thank them because this is a huge effort, not only me, but this is a real family. When I say family, is not to look nice. It's really people working real hard to make this happen every day. And obviously I have a personal love for Australia. I was there in a sabbatical last year. I spent a couple weeks in Australia doing some crazy adventures. So I really love Australia. I hope I can go back. I hope I can visit Melbourne and other areas that I did not have time. And I'm open for questions. And thank you so much for this opportunity. I really appreciate it.
Thank you very much, Renata. That was a very, very nice presentation.
You're welcome.
Do we have any questions?
Yeah, it's Vicky Marshall here. Thank you, Renata, for your fantastic talk. That was really, really good, really enjoyable. With regards to the Twist probes, what do you think? You've shown the increased coverage. More uniform coverage, which is fantastic. So, what do you think? What features of the Twist BioScience probes is behind that? Is it the manufacturing process, do you think, and maybe Jeremy can comment, or the fact that they're double-stranded probes? What do you think is the key?
I think the key is the company founder and the fantastic R&D, Emily. But I agree. I think double strand is always like the best way to go. I always thought that, and I'm very glad that you mentioned that. I do not know the secret sauce. People think I know because I spend so much and I always use so much of Twist. And some people think I work for Twist. I'm like, no, please just say that I work for CHOP and I have a pen appointment as a professor. So I'm like, no. But anyways, I really don't know. But I think there's a very interesting dynamic for the way they produce the probes. And I will rely on Jerry to explain more details about the chemistry behind. But definitely the double strand is very powerful. And I say that now with these new adapters that are shorter, they are so strong in a way that the efficiency for the ligation is fantastic. I think that's a perfect combination, but I will rely on Jeremy for more secret sauce explanations.
Okay. Thank you.
You're welcome.
I have a question as well. Renata, I was actually very impressed for the amount of work, but also for what you say that is important that I think as well, which is honesty and transparency. So I really appreciate that. And this brings to my question that was, how do you do it to join all these different people together into a project? Because it's absolutely huge, no? It's BioBank, it's bioinformatics, it's NGS, it's translational. What's the secret? Is a leader or is an institution or how does it work?
I don't know. Oh, it's a great question.
It is. It's a tough problem.
I think it's the patient. I think the patient is the answer. Because again, when we get an elevator with multiple really sick children, right? Every day, we must have a different, way to see that. And I feel my team is extremely engaged with that. I'm very passionate, basically, right? Sometimes too dramatic, but they treat every plate of DNA as a human being. That's the first day in their training with me and my lab is you must treat this DNA plate as a human being. It's not a plate of DNA. It's a person. There's a person behind as well, waiting for a result, waiting for an answer. A parent that probably lost 3 kids. I have families that I see that lost 3 kids with rare disease. So you have to be very, very sensitive to understand that. But I have to say that I'm very lucky to have a fantastic PI as a leader. And he's also very excited. He's a physician, he's a MD, PhD, and he see patients until now. So he still spend time with patients. So again, we have a combination of very good individuals technically, but all of us very motivated by the mission of the institution. So, which I think is the answer for you is the patient.
Which I didn't expect, and again, it's super nice. Thank you.
Sorry, Vicky, before when I was trying to answer your question, I was on mute, my apologies. So
That's okay.
the answer to your question is that it's based on a proprietary technology that essentially enables us to print our oligos with really, really high fidelity, basically extremely low error rates. And then on top of that, the manufacturing process and the algorithms behind and the AI behind how we generate the balancing of our probes, and then the PCR after that to amplify those probes to maintain that evenness and uniformity is basically how it works. The special sauce cannot be given away, but that's essentially the background.
That's great. Thanks, Jess.
See, Jeremy, I did not compromise anything here. I really don't know the secret sauce.
I don't either. All I know is what I'm allowed to say.
I would love to know, but I don't. I have some clues, but I don't know.
Is there any other questions?
I always gonna ask questions, so if anyone... Anyway, I'm gonna ask something, Renata. I'm sure you've been asked this many, many times. Until when the whole exome, whole genome is just pressing a lot. Well, sorry, I didn't introduce myself. So I work in a core facility, a genomics core facility in a cancer institute. So all I see is cancer. And of course, in cancer, there's a lot of pressure for whole genome rather than whole exome. How do you see that going?
Again, I'm very concerned about genomes. Maybe because I've seen, I don't see much cancer. We have cancer, but not much. But again, we have so much issues, even in cancer. If you think about it, you have fusions. You have so much different things that not necessarily a classic whole genome short reads is going to resolve you, right? We can rely a lot in bioinformatics now because we have done a fantastic job doing rearrangements with softwares now. Like we can see structural variants now, and which we couldn't really see it. But short reads is the limitation. Doesn't matter how robust is my bioinformatics pipeline, I still cannot resolve many things, even in tronic variants that we have no idea how to classify. So I still see a problem. And again, coverage, right? And in cancer, we run when we do cancer, we run minimum 100x coverage. Okay, so exome, it's cost effective too, and we can run 100x for less than $500 now. I can do that. If I have to really bump my whole genomes 30x, even with the newest instrument I have, I need a little more. I need at least 50x, and then it's very cost prohibitive for diagnostic now. I don't see that really changing yet, but yeah, it's happening. It is a trend, but I see that happening much more for, not somatic, but actually, germline, not really cancer related.
So Renata, you run some whole genomes with a Twist library prep, correct?
Yes, I do.
Do you ever run low pass with exome at the same time?
We do not. Thank you for reminding me that. I forgot to say that. Guess what? We love this adapter so much. Again, I don't work for Twist, but I really love the adapters. That I was running whole genomes with other vendors in PCR free, and even PCR free, it was like, eh. And then one day I came and I said, you know what? Let's run library for Twist without capture and do whole genome and sequence. And then I emailed Brian. Brian is someone in Twist, and he said, that's a good idea. And here we go. I run all my whole genomes with Twist, library prep, no capture. Duplicate rates are basically nothing. I have 1%, so it's not even something that I care for. And I have done some in parallel, but the reason we do whole genomes, it's because, again, we have some constitutional ones that we believe it's rearrangements, and that's why they're doing whole genomes. It's a very expensive thing. We have also pipelines that we run array first, we do the microarray first, and then last scenario, we go to a whole genome. Did I answer your question? I don't know.
Yeah, you did. I was just wondering if it was a technique that you were employing, because some labs do that these days.
Yeah, no, I don't do that. But when I do, again, I do whole genomes, but I do that with, again, the balance of like the real question, making sure the disease is very well like characterized, and if I'm asking the right question, that's when I really go into the genome. But obviously they are challenging because of the analysis.
Okay.
Do we have any more questions?
It's Vicky Marshall again. I have one more question. Renata, you mentioned extracting DNA and RNA from Ffp blocks. I'm just interested to know, excuse me, interested to know what what you use for that.
So we have done tons of different things, right? So we have successful extractions with QIAGEN, but we have a fantastic success with Promega kits.
Okay.
So our preference for challenging samples, it's Promega.
Okay.
But our blood spots, the challenging ones, they were validated for either Promega or Agincourt from Beckman Coulter. So all this technologies pretty much are validated in both different methods, just as a backup in case one vendor goes, in shortage of like reagents, we have a backup plan.
Okay, great, thank you.
You're welcome.
I also just wanted to say that we share a few things, and one is a very needy dog, which I also have, so I understand that. And what was my other one? About being brave. I really like that comment. And when you were talking about, your liquid handling robotics, I thought it was really interesting because I actually developed a liquid handling robot phobia over my career. So I really can relate to your comment about being brave. So thank you very much for your talk. And it was really good to hear about not just the technology, but, the I guess, work and motivation and ethics, et cetera, behind it. It was a really important part, obviously, of research. So, and you don't often get to hear that side of things. So that was great. Thank you.
I appreciate it. Well, I feel that people need to feel motivated many ways, right? Yeah. So the team is excited, is happy, and they are engaged because we make a very good, vibe in the lab. Yeah. People happy work, they work better, yes, and they are much more productive.
Yeah.
And they claim that I act like a mother in the office and a bad mom in the lab, because if I step in the lab, if something's really wrong is happening, I'm like, don't say that. It's the reason is you have donuts every Friday is because of that, because I want people having sugar and happiness. Yes. Thank you. And another thing is, I appreciate your comment. I don't feel scared about automation. If you need any help, let me know. I used to be completely like desperate just to see the arm of the robot move like. And now I have 4 robots doing high throughput sequence, library preps at the same time. And I do RNA-seq low input, 1 nanogram, 10 picogram, and people think I'm crazy. I'm like, no, we are going to do it, and we do it. And so again, it's I feel your phobia. Don't get me wrong. Just to share one more thing, and I'll leave you alone. During COVID, my team were like stopping work because we were hit very bad in the United States. We lost many lives, unfortunately. And in a contingency plan, I was involved to work in a COVID. So I had to come to the lab, and they said, you have weeks to create a test efficient enough to doing high throughput in a robot. I'm like, oh, sure. Come on, piece of cake. Not really. So imagine I'm out of the bench for many years, but I still go to the lab sometime. And in a month and a half, we submit an FDA EUA product that is running, and we are running nonstop in our 96 robot COVID test. So again, be brave, because again, it's very challenging, but we face CDC and we face FDA, and we have a beautiful test safe for COVID now in our robots. So just again, another encouragement that, and I was operating the robot. So it was a lot of chances to not work well, because it was me, but it's just working. So it's very good. Just be brave.
Fantastic. Thank you.
You're welcome.
All right, thank you. Thank you very much, Renata, for the presentation, and thank you for your attendance. I hope you've enjoyed this morning discussion. We've planned a few more MPCCC Tech Talks in the coming month. The next topic is custom and Amplicon-based assays. The two different designs for next-generation sequencing. Daniel Thompson from Monash Health has a great deal of experience with both approaches, and he'll share with us his perspective around these two assay designs in the next talk. So I hope you can join us again in August. If you haven't already, please sign up to the MPCCC newsletter or follow the MPCCC on social media. It's the best way to keep informed about upcoming Tech Talks and MPCCC events. So again, thank you very much, everyone, and have a great rest of the day. A great evening. Renata, thank you for spending your evening with us.
Thank you,
Thank you
Renata. Thank you, everybody, for joining.
Thanks so much. Thank you.
Bye.
Bye. Bye.
Bye.